Well, hey, forensic friends. Dr. G here. So, we're going to do a little discussion about forensic entomology. So, how many of you have ever watched the TV show Bones? Raise your hands. No, don't really. This is a video. I can't see you. Just whatever. So, if you haven't watched Bones, well, you have to because it's important to this class. Dr. Jack Hodgins is the slime and bug guy. And one of the first things he does when he's at a body at a crime scene is he begins looking for maggots. And he always gets excited when he finds different kinds of maggots because there's a lot of information that the insect activity on a body can tell us. I've got a little PowerPoint spiel I'm going to go through here. We'll dim the lights and turn the screen down. And I'm going to post this PowerPoint as well so you can kind of play along with it or whatever. But before we get into it, there's a couple of vocabulary terms and ideas that are important, and I just want to go ahead and throw them out. The first is that not all insects are as useful for forensic identification as others. There are different classes of insects that go through different kinds of molds. Now, pretty much all insects have to go through molds. Spiders, for example, well, they're not true insects regardless. Let's see what else are insects. Crickets. They go through the stages where they are hatch out as a little nymph, not as a maggot. And the nymph is not fertile. And it goes through a series of molts where it sheds its exoskeleton and it gets bigger and bigger until finally it reaches its last molt and it's sexually fertile and it's able to actually mate and produce eggs or boys and girls, you know the story. And those aren't as useful for forensic identification timelines of death as other insects. Now, insects you might be more familiar with, things like butterflies. I used to work on fruit flies. Things that have a worm or larval stage, they're a maggot, and then they undergo metamorphosis. They actually form some kind of pupa or a chrysalis, and the maggot, the worm, will change complete shape, going from a worm or caterpillar into a gorgeous butterfly or a maggot into a fly. Well, this class of insect and this class of this sort of lifestyle is referred to as being holometabolous, meaning that it has what we call in the insect biz a complete life cycle. That means it starts out life, as we all do, as an egg. The egg gets fertilized. We have the embryo, and then we have a series of stages that are known as the larval stages, and these are referred to as instars. And depending on the organism, there may be 3 or 4 instars. In flies, there's first instar, second instar, third instar, and these are larvae that will look like that, and then they will molt, and they'll get bigger, and then they molt, and they get even bigger. So we got mouth parts, and they have little bristles on them, and these little sort of breathing tubes at the back end. After the third larval molt, at least in flies, they undergo the next stage, what is known as pupation. Now, pupation is when they form what's called the pupae, or the pupal case, the pupa. And during pupation, something really quite magical happens. It is known as metamorphosis. It's very much like a transformer in that, not to get too much into the detail, but it's actually a really cool molecular genetic idea going on here. They don't change cell number. They don't carry out any more cell division. They just carry out cell rearrangement. And they go from being this maggot shape to being a winged creature. And so after metamorphosis, this pupation, and they then will pop out of the pupal case in what's known as eclosion, and they are then fertile adults and ready to carry out the life cycle again. Now, what makes this critical is that this is a pretty much set in stone life cycle. The insects that we're going to be looking at, they are laid as fertilized eggs. They undergo embryogenesis. The embryo hatches out as a first instar larvae. They go through larval instars, and it's a very precise set number. They go through pupation, metamorphosis, they eclose, and you got a fertile adult. And we can actually time these things. Now, not to the minute, but I will tell you that flies under controlled conditions, their life cycle will be so synchronous that you can have several hundred embryos or fertilized eggs laid, and 10 days later, you're going to have pretty much within that day, and probably within a range of a few hours, all of them may close and come out as flies. So it's a pretty cool schedule. Now, that's a critical thing. Now, another thing that's critical to think about with insects particularly is they are not warm-blooded. They're not able to maintain body temperature. And so because of that, the temperature at which they are raised in which they're experiencing, is going to control their rate of development. So these are going to be temperature dependent times. Now, we can take average temperatures over the period of when we believe the samples have been, after the eggs have been laid, and you can develop a average temperature that will give you an average time for the development that it takes to get to whatever spot you happen to pull somebody off. So that's a critical point, and really it is a critical variable. So the temperature dependent times are something that you have to actually have some experience. You use the best weather data you've got. And as we're going to see, depending on what kind of environment that the body is in, that is it buried? Is it on the surface? Has there been rain? It's going to come into effect, and these are various different variables we have to put into play. Now, we're going to have a little worksheet kind of thing that I'm going to give you that is mainly looking at temperature and figuring out time from that. But we're going to see there's a whole range of variables. Additionally, we can use insects to look at the kinds of material they're feeding on. Sometimes they may accumulate different kinds of toxins or drugs if the drugs are in the body. So there's a lot we can determine by looking at the insect population on remains. Ooh. Okay, let's do a little bit of PowerPointing and see where we go with this. Oh, here we go. I'm going to bring the screen down, and we'll kill the lights a little bit. So, we're not going to take dead meat and put it out in the parking lot. It'd be kind of cool, but really quite disgusting. I'm not into doing that. Another thing just to keep in mind, there's another thought I had. How many of you have dogs? Again, don't raise your hand. Have you ever noticed you're out taking your dog for a walk, or you're out on your own lawn and your dog takes a poop? And what's really kind of interesting is that within moments, seconds after your dog has done their business, there will be flies buzzing around it. Flies drawn to poop. And you might say, where the hell do them flies come from? And that's actually a good question. They probably are hiding in the grass, just sort of waiting and just hanging out. And when they get the smell of fresh poop, They are attracted to it like flies are attracted to poop, which is what they're doing. And if you're not really paying attention to this and doing some experiments, it's easy to sort of, and naively think that they just sort of are magically appearing. And this is one of the things that is an interesting little point, is that back in the 19th century and prior time, the idea that life was just spontaneously coming about was something that was thought of, and that's known as spontaneous generation. And one of the experiments that demonstrated spontaneous generation, and by the way, it's not true, was you take a piece of meat and you set it outside and you come back and a few days later and you'd see maggots crawling on the meat. And you would make the obvious assumption that life begat life, that, some sort of life force that's in this meat is generating these maggots. Okay. Pasteur and others actually demonstrated that spontaneous generation doesn't work by taking the meat, putting it into a box that had a screen over it so the flies couldn't lay their eggs, and lo and behold, no spontaneous generation. So what's happening is the flies are being attracted, in insects in general, to the cadaver, to the dead body. And they're laying eggs there, and then they are allowing those eggs to hatch into larvae. And the larvae then start crawling around. And pretty much, if you've got a dead cadaver, necrotic material, flesh, laying out on the surface outside, within 10 or 15 minutes, there's going to be fly activity on it. They're going to be laying eggs on it. So that's how we can begin to use, for a time of death, different kinds of entomological markers and things. So this is a PowerPoint that I have lifted. It's interesting, and we're going to go through some of it. Some of it I'll let you read through. If you're interested in a cool job as a forensic entomologist, that's a neat thing. You probably are going to be studying in graduate school a lot about insect behavior and insect development. And so, as one of the things they're saying here is that maggots can respond to various drugs and actually speed up or slow down the insect's development. But in general, a forensic entomologist's job is going to revolve around identifying insects that are at a crime scene and understanding the different life cycle stages, how long it takes to get there. You would collect or preserve evidence so you can actually demonstrate this in court. You can determine an estimate, anyway, for the post-mortem interval, as it's called. That is, the time between death and the discovery of the body or the discovery of the evidence. So insect type, insect stage, life cycle stage, the weather conditions, the location, the condition of the body, is it been buried, is it on the surface? These are things that you would do. And then eventually you end up testifying in court explaining all of this stuff. So most insects that are used are really in two different kinds of what are known as orders. You have flies and you have beetles. And when you begin studying the kind of changes that a body undergoes and the insects that are on it, we have what's known as a succession. That is, some species of insect are going to start very early, and others wait until the material is been dead for several weeks, starts to actually begin to rot. And you can find a range of insects on this based on how long the body has been exposed. One of the first guys to arrive on the scene, here, let me get my little red pointer, is the blowfly. And blowflies, they are going to be present on a corpse very early and hang around for a while. As the corpse begins to deteriorate, beetles start to come in, and then there's different kinds of flies and beetles as the body continues to decay. So not to be overly graphic, but we have a number of stages that are being driven by insect activity. One of the things, again, if you've seen on bones, and it's actually a standard tool, is you can take an animal, and if you want to get the bones, the skeleton, you put it into a box with a lot of beetles, domestic beetles, and they actually begin to chew away the flesh, and they will leave you with an absolutely beautifully stripped skeleton. So we've got the fresh body. It begins to bloat as gases begin to accumulate, and then after a while the physical body begins to actually start to fall apart. It begins to decay. After decay, the body begins to dry down, and then as the flesh has been stripped, all the squishy parts, you're left with the bone. And the bone is pretty much not going to get messed with. I mean, there are animals that can take it and things, but bone is not going to be broken down by insect activity much. So you may have, I think we probably talked about the body farm, where they actually take contributed corpses and put them out into different kinds of conditions and areas and able to observe this. And we have this kind of information. These are little piggies. Thank you very much. Fresh body, that starts at death. Flies begin to arrive, and the temperature of the body is going to begin decreasing. Another standard thing that a coroner will do is take internal temperature of the body as a way of determining the time of death. And so this also depends on the external environmental temperatures and breeze and things. But there are sort of equation cheat sheets you can use to actually determine more or less the time of death. The degradation of all the molecules, the complex molecules in the body, is beginning pretty quickly. Bloat starts to happen after a day or so. The bacteria that are in the gut and maybe enter into wounds, they begin to produce gases. The temperature of the corpse actually begins to go up because of all the bacteria activity. You're still going to have lots of flies. Decay is actually a fairly long time. Gases are subsiding. The body is no longer really intact. Things start to leak out. The bacteria and the maggots are breaking through the skin. You get a lot of maggot activity. It's going to smell pretty darn bad. Larvae begin to pupate. This is actually after a week or so. The corpse is going to dramatically shrink as the moisture begins to move away. And again, this is going to depend on whether the body was placed in a real dry environment or a real wet environment. But these are things you can begin to actually understand. Post-decay, you're going to be left with the carcass, with the hair. Skin is going to be pretty tough to break down, so that's going to be left around, and bones. The fly population actually changes a bit, and you have other kinds of insects and different small creatures coming along. There are beetles that are present here, particularly in dry environments. The beetle population, they start to really pick up. They take a while. They kind of come in near the end. They ain't big on fresh meat. So here we go. Lovely. A hide with maggots all over it. And can we zoom in? Not that we want to, but you can see there's maggots of different sizes. There's a guy there. Little tiny guys, real big guys. These are different larvae. All right, so the dry stage, if the corpse is in a wet area, say in a river bottom or near the shore, it may never actually hit there. The maggots will stay there, and the hide beetles never really get a chance to strip things down. But in dry conditions, the corpse is going to be reduced down to the skeletal stage. So you only have bone and hair remaining. All right, so we had this idea of the post-mortem interval, the PMI. And there are a number of variables and the PMI. And I wish I could write them down, but let's just see what we can do here. Tips? No. All right, anyway. I was going to highlight this, but I can't do it in Google Slides. Weather data is a critical tool because the temperature and the rainfall is one of the really critical variables. So you collect weather data related to the daily temperatures, the highs and the lows, and the number of hours at the different temperatures. And you can get this from different sources. From NOAA has very good regional areas, and you got to make your best guess of what's going on in the body specifically. Now, one of the things that's interesting here, I kind of think of this from two different sides. If I'm the defense attorney, how am I going to go at you? And if you're the prosecution, if you're witness for the prosecution. If you're in a low area, the temperatures might, in the evening, get cooler. If you're in a high area, a little bit of a ball's upper spot, the wind can be blowing a little bit differently. Temperatures may not drop as much. So there's a lot of things that are a little bit squishy about this post-mortem interval. The time of death determination. Was the body wrapped or in some kind of material that would slow up the flies from finding the body? Was it wrapped up in a garbage bag? Was it wrapped in plastic? Was it buried? Are there other insect species around that might have affected the guys that are being collected? Insects can be preyed upon by other insects and things. Are there drugs or poisons there in the body that also are going to affect the larva? So if the guy is doing a lot of speed, hey, that's actually going to have an effect on the insects. So the decay rates then are variable, and the body farm is a critical place to study this kind of stuff. You can break down the variables into temperature, access, and then depth of burial. So easiest is if you've got a body that's just laid out on the ground. That's going to be the simplest. Other factors that you have to consider would be things like embalming agents. If the body had been treated in some way, things like adding lime, adding insecticide as a way of kind of spoiling the forensic investigator's ability to get information. Lime is going to begin to stabilize the body a bit, keep it from rotting quite as fast. Another critical problem, of course, would be scavenging animals. You get coyotes in there, yanking pieces out. So, ain't none of this is cut and dried. So here's an interesting little set of data points. In terms of time of death and how you can determine it from a variety of different kinds of characteristics. Now, this is not insects. This is just taking basal body temperature. If the body is warm and not stiff, which is, of course, something that's called rigor mortis. Rigor mortis is going to set in typically after about 4 hours or so. It's a physiological condition that fixes the muscles in place. Now, prior to about 3 hours or so, the muscles are going to be movable. You can actually bend the body around. So if the body is warm and not stiff, it's probably less than 3 hours. If it's warm and stiff, it is now in the stage what is known as rigor mortis, between 3 to 8 hours, depending on the ambient temperature. If the body is cold but stiff, that's probably between 8 to 36 hours. After about 36 hours, the body begins to decay sufficiently that rigor is no longer an issue. The muscles are not able to actually maintain their structure at all, and you start getting into bloat and stuff. So if you're cold and not stiff, you've probably been dead more than 36 hours. So the forensic entomology biz is typically focused on violent crimes. Determination of the post-mortem interval. Now, other times that a medical or a forensic entomologist might be called in is if it is a sudden death that's particularly in a remote area. Traffic accidents actually can be this. If there's no obvious immediate cause, did the person have a heart attack? How long have they been in this car wreck? So, like, you're driving in a remote part of the country and there's a car that's crashed. You can begin to make some investigations about how long that body has been there, and there's ways of trying to do your best to interpret whether it was accidental by somebody having a heart attack or falling asleep, or was there another car involved with it. So, I guess the possible criminal misuse of insects. All right. So the phylum, the general group of animals we're talking about, are known as arthropods. And arthropods, or the arthropoda, can be broken down into two major groups. Arthropods like crabs and shrimp and spiders, and insecta. So they all have a hard exoskeleton, and because of this, the hard exoskeleton has to be shed if they're going to grow. So we've got a variety of different kinds of insects. There we go. All right, so we can determine the PMI, the post-mortem interval. Some of the things you can determine with this is the movement of the corpse. If the body was killed inside and then a few hours later dragged outside, there's going to be some discrepancy between the temperature and the insects. Potentially the manner and cause of death may be useful. You may be able to associate suspects with the death scene. Certainly you can take the larvae and grind them up and actually begin to identify material in there. DNA, maybe. The bloodsuckers are actually going to have some of the material there. And it may be actually of higher quality than the tissue on the organism, on the remains. Although I would think that a little bit of cracking open a bone and getting some bone marrow out would do you just fine. But regardless. So, as we say, in order to get the post-mortem interval, you got to take into consideration the environment, because these insects are not warm-blooded. They are their development is going to respond to the temperature, which is a very weird thing for us to think about. But imagine that, pregnancy is 9 months in humans, but if you're living in a cold environment, it might go longer. Now, of course, just so you know, that's not the way it works. Just didn't want to make anybody confused. People in Alaska still have children after 9 months. But with insects, development is going to be perturbed by cooler temperatures, and it's going to be sped up by warmer temperatures. So, the time of day the death occurred is a critical point. Did it occur where you get a blast of sunlight and warm weather for a few hours, or did it occur at night? The time of year is critical because that's going to have an effect on the overall temperature. If the corpse is in water, probably not going to have a lot of insect activity. If they're in the soil, you're going to get different kinds of things depending on how deep they are. One of the take-home points here is there are going to be some conditions, either timing-wise, that is the seasons, or treatment of the body, that, whatever you might, the perp did to it, that are going to make it so forensic entomology may not be able to give you any information. All right, so these decay events very quickly begin to send out signals, primarily in the form of smells, that say there is a dead body over here. The flies are the very first to hit. The blowflies are flesh eaters, and they are first to the trough. And just like I was saying with your dog poop, flies can get there within minutes. Now, depending on the kind of area you're in, like if it's really sandy and there's not a lot of grass cover for the flies to actually hang out in, it may take a while. It may take a couple hours. But that smell will travel. And when they know there's dinner, man, they're going to come from miles around. It's very possible that the flies will arrive before you have actually completed a crime. That is, if you've opened up wounds and you have blood, that's going to bring the flies around. Okay, so the fly life cycle becomes a critical point. And there's a paper I'm going to be giving you that has an exercise that gives you some of the times, and this is how you begin doing it. The females lay eggs, and they typically lay the eggs in exposed body openings. They don't lay them on the surface of the skin. So you get into ears, the nose, the mouth. Depending on the coverage, you could have them in your anus and genitalia. Oh, God. Man, I'm a molecular geneticist. I work in sterile environments. The last thing I ever want to do is say the word anus and genitalia. Hey. All right. The different kinds of insects do different sorts of things. Some lay eggs. Some actually lay larvae that have already hatched. And the mom is going to actually begin digging into the body and sort of making it so the body openings are really good spots. Some flies actually can chew in. Blowflies have spongy mouth parts. They actually have to have material that is somewhat dissolved before they can actually kind of suck it up. So that's why they put things, start laying stuff into these orifices, as it were. So here's some blowflies. There's the dude's spongy mouth parts right there. Yum. They have a kind of a cool iridescent blue-green to them. Blowfly eggs are about half the size of a grain of rice. And generally, when mom lays eggs, she doesn't lay them one at a time. She'll just actually sit down and just start shooting them out. And so you end up with these clusters of eggs. And you can see them. If you know what you're looking for, you absolutely see them. So if things are colder, you're not going to have as much. You can even have them in some of these, more hidden locations. These flies actually can get into all kinds of weird places. Now, the eggs for blowflies are going to hatch in a few hours. And it depends on how rapidly mom is laying them, because pretty much she is fertilizing them as she's laying them. They actually can hold sperm. And so if they're laying fast, that's actually a really synchronous population. And so you can begin to look for the hatching and the timing of the hatching as a way of measuring when the fly first set down. Let's see. You have a larvae that comes out, and to be honest, to determine whether it's a first instar larvae or a second instar larvae, you really got to actually know your stuff. You have to have a pretty good microscope. And with flies, I usually look at it from size. The third instars are pretty big. First instars are teeny little guys. There's also a variety of bristle patterns and these breathing tubes called spiracles that are also involved in the identification. So here's some maggots. Yum. And they're going to all hang together sort of as a little family as they feed on the corpse. And they start to move down. And so what's happening is they actually are secreting digestive enzymes to make it so the flesh begins to dissolve, and then they can more easily suck it up. Oh, yummy. All right, so blowflies are going through these three instars. I think of instars as a little bit like, if you've seen the movie Alien. If you haven't, you should watch it. The alien goes through a series of life cycle changes. Now, in flies, it's not that dramatic. They look like little maggots. Then it's a bigger maggot, and then it's an even bigger maggot. And unless you really know what you're looking at, they all look pretty much like maggots. Hey, one maggot looks like another maggot. In the alien, you had the egg that went to the facehugger stage, and then it went to the chest burster stage, and then it underwent metamorphosis and became the big mama. Whoa. All right. So blowfly larvae, they're not quite this dramatic. So they go through to the third instar, and depending on the environmental conditions and to a degree on the species, it can take different times. The number of larvae present too. If there's a lot of food and there's not a whole lot of larvae, they can go a lot faster. If they're having to compete, they may end up going slower. So then they undergo pupation. And pupation is actually going to be where they are going to find a place that's kind of out of the goo. And they burrow into the soil. With flies, with fruit flies, they actually will crawl up on the side of your garbage can, something like that. And that's when they go into the pupal stage. And that takes a while to actually carry out metamorphosis. Now, with caterpillars, they spin a cocoon. With most other holometabolous insects, it's nothing nearly as dramatic or beautiful. It's mostly they take the outer exoskeleton and just kind of make it crusty. And inside is where they actually begin to change shape. The pupal case is going to be made of chitin, which is basically like a shrimp shell or a crab shell, and they can hang around for a very long time. Okay. So this is the blowfly life cycle. At 70 degrees Fahrenheit, each stage has a known amount of time. And we've got mama here laying eggs. The eggs take, at 70 degrees, 23 hours. You have a first instar. Aw, it's a little baby. 27 hours later, it molts. 22 hours later, it molts again into the third instar. And then it's going to hang out as a third instar for a while. It actually does a lot of feeding there. 130 hours, how many days is that? I don't know. It then pupates. You can see how it kind of shrugs itself in, hardens up the outer shell, and then the inner goo, the inner cells, actually begin to undergo metamorphosis. Metamorphosis takes a while. That's the longest period. 143 hours, and then they emerge. So by looking at the kinds of stages on a body, you can begin to make a time index now. So, let's see. We've got these different stages, and the larvae go through their instars, and then the larvae develop into pupae as they bury under the ground. The adult fly emerges, and it's going to take between 14 and 16 days from egg to an adult, depending on the temperature and the humidity. So we can determine post-mortem interval this way. You first identify the species of the bug. That way you know what its life cycle time is going to be. You are going to document the kinds of larvae you see. You're going to see little tiny guys. What's the oldest larva you see? That's the longest one there. Figure out the instar. And from there, you can begin to determine the post-mortem interval estimation. So you have what's known as the degree day, which is the number of the temperature off of a baseline media, say at 70 degrees. How many degrees above that or how many degrees below that? Determine this way you can begin to incorporate a variable, the temperature variable, into the calculation. Now, the life cycle is going to give you an estimate of the time that the corpse has been in the location. Just as a little point, the post-mortem interval does not determine the time of death. That actually is established by the medical examiner. What you're going to do is provide them with data that they can use to make the best guess for time of death. And so, post-mortem interval and time of death may not always be the same. The post-mortem interval is the time that the corpse was exposed to the environment that allows insect activity to begin. And things like being sealed up, closed windows, being in a bag, very cold temperature, being buried deep, that's going to change the PMI from the actual time of death. But it's good information. All right, we're going to stop here, and what we'll do is I'm going to give you this assignment and let you work through it, and we'll meet back up I'll probably drop another video this week to go through it a little bit, and next week we can zoom a little bit to talk about what's going on. So, that's how we can begin thinking about bugs and death. Oh boy. All right. I'll see you in the Zoom ether. Come on by, don't be a stranger. When we start getting into some forensic DNA stuff, we're probably going to, in small groups, gather and extract some DNA from ourselves. And actually, we've been doing some work this week, getting some information on how we can actually detect some of these DNA fragments that are used in CODIS determination. And it's actually working out fairly well. I'm looking forward to trying it with a population of you guys. All right, I'm Dr. G. I'm out of here. Peace.